everyone for coming. Today we're going to hear from Kat McCollough and Erin, who are students at Mission College. They're here because Professor Jean Replicon from Mission College developed this summer research internship with us. And when I say us, I mean SFBBO and specifically Coyote Creek Field Station, our bird banding station. And I do want to say Jean put in a huge amount of work in developing this program and working with the students. I'm really excited that she can come watch this. And this program came about because Jean is part of a group that SFBBO is also involved with that is working on developing something called SOAR, S-O-A-R, which stands for Student Opportunities in Avian Research, which will help connect educators and students with experiences at the bird banding station. So Jean was there and we were there and we started talking about ways to connect and Jean just went for it, which is so fabulous. So this summer research program that Kat and Erin just did is a pilot project for SOAR. And I think you'll see why we're very encouraged by how it went. We definitely lucked out having Kat and Erin as our pilot students. I won't take too much of your time introducing them because they have an introduction for themselves, but I will say they dove right into this experience with wonderful enthusiasm, and they committed so much time and energy and so many very early mornings to this. I started thinking of them as sort of good luck at the station also because we got a lot of cool, unusual birds when they were around. But with that, I want to make sure they have time to talk. So take it away, Erin and Kat.
Thanks, Katie. And thank you everyone so much for joining us. Welcome to our presentation, Learning to Become a Scientist. Before we really get started, we want to share with you the sounds of the station at about 6 in the morning.
Hi everyone, my name is Erin and I'm so thankful to have had this one-of-a-kind opportunity to work alongside the amazing staff and volunteers at the Coyote Creek Field Station. I never thought I would be working with birds or studying their populations, but here I am learning more and more about them every day.
Hi, my name is Kat. As the bio says, I'm actually more of a mushroom and a plant person, but I'm really glad I didn't hesitate to throw myself into this opportunity. There is so much more to birds than I would have ever imagined. And like Erin said, everyone at the station was really amazing. This is a first for both of us, interning and doing field research. We were both very fortunate to be selected through the HSI STEM internship program through Mission College.
I felt somewhat prepared to work with birds because I've been surrounded by animals my whole life, including chickens, thanks to my mom, who's probably out in the audience right now, and she's a veterinarian. Now, chickens aren't very similar to the little songbirds that we worked with at the station, but I have rescued some sparrows that got into the house, which is about the extent of my experience with wild birds until this internship.
So I've had birds before, kind of. They were two giant blue and gold macaws, and they weren't really friendly. I didn't actually really like them very much, but they did give me a lot of respect for birds that should probably not be pets. This is my first experience with bird banding or doing anything with birds really. And it was really exciting seeing so many species that are completely new to me, but that live all around me.
Like I said, this is my first experience working in the field with banding birds. And actually, when I first heard about the station being located near Coyote Creek, I thought we would be working with shorebirds. And on our first day, it became very apparent that we would not be working with shorebirds at all, but almost exclusively songbirds. It was also interesting to see the diversity of species living so close to well-developed areas like McCarthy Ranch and the Water Treatment Center. I was also impressed by how dedicated everyone was to ensuring that the birds were taken care of and accurate data was being recorded. No one was scared to ask for help, clarification, or a second opinion while collecting data to ensure it was all accurate.
So I've actually lived in McCarthy Ranch, or not McCarthy Ranch, but in Milpitas my whole life. And I've even worked at McCarthy Ranch before. I've gone hiking in the area, but I had no idea that Coyote Creek Field Station existed. I think one of my favorite aspects over the last couple of months has been the nice, awesome, quiet nature talks and walks. There aren't a lot of places I spend time where I don't hear any humans. Nowhere else in Milpitas do I see such a concentration and diversity of birds either. It makes me realize how important the station and the strip of land it's on is. I came into this internship with a pretty open mind about as far as what kind of birds we'd be working with, but I knew I wanted to do a project that was relevant to California's issues, either something to do with the climate, the drought, or the fires.
When we started at CCFS, we had to get into the routine of the banders who run the station. So twice a week over the summer on Saturdays and Sundays, we arrived at the station around 5 a.m. to observe and learn firsthand from the banders.
The main goal of this internship was to introduce two completely new to the field students to some real life experiences in wildlife research.
One of the first skills we observed was something called sculling. Sculling entails looking at the bird's skull formation, and it will give an indication of its age. For some birds, however, trying to determine the age by skull is impossible. A unique example is the woodpecker pictured here on the left. The woodpecker is a special case because its tongue actually wraps all the way around its skull. But for other birds, it can be because they have dark skin or thick plumage, making it challenging to see the skull. And just a side note, both these birds are completely fine. The banders just use some water to part the feathers to make it easier to see the skull.
One of the smallest species that we catch is called the bush tit, and on average, it weighs about 5 grams, which is slightly less than the weight of a quarter. When you're holding them, you can hardly feel it, and they are my absolute favorite species that we interacted with. One really cool thing that I got to see was that several of the female babies with their eyes transitioning. The females, like here, have lighter colored eyes, but male and baby push tits have dark eyes. So as they're getting older, if you come at the right time of the season, you can see that transition period. Bush tits actually visit my lemon tree at home, which I did not know before. It's really cool that I can identify them now. Before they were just the cute little chibi birds that came around all the time. So here we actually have a video of Julian, one of the banders at the station, explaining the proper way to hold and handle and release the birds. This was actually Erin's first bird release ever.
My index finger and my middle finger are around its head, and then my other fingers just cradle the rest of the body.
You want your non-dominant hand.
Yes. And
is there a safe way to transfer it, or
yeah, I'll show you in a
Okay.
sec. When you release the bird, put one hand underneath and then just open your other hand.
Okay.
That way, if the bird doesn't want to fly,
Releasing birds was definitely the best part of the experience for me, I think.
All right, so I'm gonna pass it to you holding its legs like this. You can just get your hand around. Perfect. Got it? These can get a little tighter. Perfect.
I'm gonna kill it.
Don't worry. I promise that will not happen.
The neck is okay.
And you're gonna put your other.
Yeah, I was very nervous for this release since it was my first one ever.
Oh. Do you encourage it or just let it sit there?
Let it sit there, and then if it doesn't fly away, you can give it a little bump. So there you go, just like that.
And then on the right here, we actually have a compilation of a bunch of bird releases.
What an opportunity.
Wow.
Don't look at me.
Oh, that was cool.
This is one of our favorite shots because you can see it flying away as a shadow.
Also ask for
He's ready.
band size justification. Recommended band sizes for this.
Ready? Whenever you're ready. Ooh. Your guy flew right by me.
All right, so we talked about how the banders collect a bunch of different data. They use things called data sheets to keep everything organized, and they assign numbers or letters to a bunch of different categories like band numbers, featherwear, molting, breeding characteristics, skull growth, and more. And it seemed really crazy to be able to remember all of it until I was actually asked to help record that data. Writing in the data sheets, for me at least, made it much easier to understand how it all works. Listening to the banders explain their reasonings helped me learn even more about the birds. And this experience also highlighted how important consistent training and data collection is for accurate records. And this is an example of an actual data sheet. Each row is a different bird, and you can see how much goes into each one. And while we were there over the summer, the volunteers and staff were working on transitioning to a digital format using an iPad.
So here we actually have a video of the old system. Erin is recording in the logbook over here. And then we have Allison, one of the other banders, recording on the iPad over here. This is a new digital system that volunteers have begun to integrate over the summer. The old system of pen and paper has been very reliable for many years, but there was room for errors, and the time it took in getting everything digitized was becoming problematic. It would have actually been awesome for us to have the iPads been up previously, because it would have saved us a lot of time in transferring data from the bird wall to our data set. Now, as soon as the birders finish their entry, it's automatically uploaded into the system. Also, when you see Katie blowing on the birds, that's actually how the banders move the feathers around to safely see the bird's skin. Coyote Creek is a really important place for multiple reasons. With the location of Coyote Creek being in the middle of a very urban Bay Area, the station acts as a stepping stone for migrating species to take a break and rest up before moving on to the next destination. Additionally, the station is located by a riparian corridor. A riparian corridor is a unique plant community consisting of the vegetation growing near a river, a stream, or any other natural body of water. It serves a variety of functions important to people and the environment as a whole. Some of those functions include preserving water quality, filtering sediment from runoff before it enters rivers and streams, protecting stream banks from erosions, providing a storage area for floodwaters, and providing food and habitat for fish and wildlife.
Besides the ecological advantage the station offers, the fact that it has been in operation for over 30 years is a major reason why the station needs support, so it can continue to collect bird data for many more years to come. This data can be used by many people for a variety of projects, and considering that birds are an indicator species, it is crucial to do long-term studies on them to see how drastic the effects of pollution and climate change can be. Also, for as long as Coyote Creek has been collecting bird data, they have been consistently training banders in how to handle and record birds, which is incredibly important to their continued conservation.
Another great aspect of the field station has been the data collected from the revegetation projects in 1986 and 1993. They learned from their first habitat restoration that birds need a lot more understory and brush plants to provide a more well-balanced ecosystem, and that totally influenced the plants used in their 1993 restoration. Despite maintenance, though, invasive plants have taken over the overflow and some of the creekside areas, making Coyote Creek a great place to, unfortunately, study the effect of invasive species on resident species.
We learned a lot about fieldwork, field research, and proposing and carrying out scientific experiments. I think one of the biggest takeaways we're both leaving with is that it's good to be adaptable. We started with one idea and ended up with something completely different. And when it's only an 8-week project, every day is precious. As soon as we started to realize that one hypothesis wouldn't work, we didn't get stuck or disappointed. We just took a step back and looked at other project ideas we had previously brainstormed and moved on. This leads into the next thing we learned, which is that every experience imparts knowledge, meaning that you can learn just as much from failure as you can from success. Overcoming obstacles is a major aspect of what every profession deals with, and failures are really just additional learning opportunities. So it's important to keep a positive mindset and not let things discourage you.
Another thing we unintentionally came across was the temptation to admit data that didn't quite match with our hypothesis. And that's not what science is about. Science is about presenting the facts exactly how you find them. With programs like R and GAM, which we'll get into more later, it's tempting to just keep plugging in data until the numbers give us something we want to see. At a certain point, you need to stop running graphs or looking for a better answer and make a story based off of facts given. When data doesn't do what you expect it to, there's usually a really good reason for it. And when working with birds, they all react differently. Each bird has its own story, and while it's tempting to generalize, that doesn't give the entire picture.
Now, you may be thinking, why are we talking so much about learning from failure and looking for answers that aren't in the data? Well, we learned firsthand with our original idea involving rhododendrons what it means to be adaptable and persistent.
On one of our first days at the station, we noticed there's a plant that grows near the creek that looks and acts like bamboo, but it's completely useless. It's called Arundodonx or wild cane, and the volunteers confirmed that it's highly invasive. Here on the right, we see our teacher, Jean, and her husband, Paul, who both helped us immensely with some of the arundo removal. And then Katie, on the right, is sitting in a nest of the arundo. You can also see the actual net we're trying to clear around, which hopefully you can see my cursor, but it's this black line down the middle of the photo here.
Now, this is a time lapse of the work that we did. The idea behind the Arundodonx project was to see if clearing out an invasive plant species surrounding a specific net would lead to more captures when compared to a net that still has Arundodonx around it. And upon doing some research, we found that other people have done similar studies, but instead of doing it over the course of one summer, they had 2 or 3 years to see results. This was the first time I realized that we may have bitten off more than we can chew because we simply didn't have enough time to see a difference.
Another major issue that arose with the Arundo project was that the particular net that we were looking at, which is actually labeled 9515A, it hardly caught anything in previous months and even the years before the invasive plant had taken over, which gave us a really small sample of data to work with. We did the Arundo removal. Another issue was we did the Arundo removal the second week of our 8-week project, meaning we would only have 7 weeks in which to collect data on the net after being cleared of Arundodonx.
And the final issue that made us realize we would need to switch projects is that we lacked the resources and manpower to really make a dent in the Arundo. Literally within a week, it had grown back taller than me. And while patches of it still remain upstream, we'll never be able to completely get rid of it downstream. And after realizing our original plan wasn't going to work out, we eventually decided to research an idea that we think has a lot of relevance and has been on everyone's mind, which is, does drought affect bird populations over time?
Our final project idea was to study whether the lack of precipitation, or rainfall basically, over the past few years will show any effect on bird populations. Our hypothesis was that bird populations would be negatively affected by lack of precipitation, meaning that we think that their population would potentially go down.
And these are some of the resources we used for our project. The first and most important were the field station's banding records. The station's been collecting data since the 1980s, and they made the data from 1993 to present available to us for our project. We realized the main purpose of bird banding at CCFS is to better understand local bird populations and their habits, then share this information with other scientists, policymakers, and locals who want to preserve the biodiversity of our region. Each bird is banded with an aluminum band with a unique 9-digit number, and all birds, new or recaptured, are recorded with the capture date, time, and the net it was caught in.
Knowledge about the migration habits of birds in the area was also really important to our project. Our data did confirm that eventually, yes, some birds migrate and some do not, but it was a great starting point of comparison for us to look at resident versus migratory versus wintering species and try and get a variety of these. After learning more about migrating birds in general and how much more affected they are by habitat destruction, mainly because they visit so many different places, we added to our hypothesis with the assumption that migratory birds would be more affected than our resident bird species.
A major part of our project was the use of a computer program called R to make generalized additive models, or GAM for short. The GAM model we used analyzed total captures per month of the 18 species we chose and compared it to total precipitation per month. GAM also summarized the data as graphs for 1 year and for all time that data was available. We also use GAM to control for net hours because if nets are only open for 1 hour, naturally there will be less birds caught than if the net was open for 5 hours.
And finally, for our project, we use data supplied by the National Oceanic and Atmospheric Administration for our precipitation data starting in 1998. Because of the discrepancy in years available for our data, we decided to use only information from 1998 onwards from the banding station. There were several months in 2019 and 2021 missing, as well as the entire year of 2020. For those years, I used information from the same field station, which you see here, the Moffett Federal Airfield, which is pretty close to us. But I had to patch the information from another website called the Weather Underground and patch all that data together. As you might imagine, precipitation numbers in the Bay Area are incredibly low, and we ended up using total monthly precipitation as a comparison value because of this. In this slide, we have an overview of the actual station map and all the nets here. When we were talking about 9515A, the Arundo Donax net, this is this here. And once again, hopefully you see my cursor, but 9515A there in the orange. And as when we were mentioning the overflow channel with the invasive species, that's all right here. About once a year, this area gets flooded, which carries down the invasive species. The riparian area is the woodland spanning alongside the creek, which is right here.
And then having the nets coded is crucial because finding out what bird was caught in what net is important to the data. Where a bird caught can show a lot of things, including its preferred habitat, where it hunts for food, and the time of day or the amount of sunlight each species prefers.
And we also just want to mention that the birders are amazing about checking nets regularly so that no bird stays there an unnecessary amount of time. And during the summer and winter months, when it tends to get hotter or colder, the birders will actually shut down the nets earlier, depending on the temperature, because birds are actually sensitive to extreme temperature changes.
This is a video of what we do at the end of every day that we're banding, and that's furling nets. The volunteers make sure to close the net as tightly as possible to make sure birds don't get caught in it when no one's around to monitor it. Yeah. It is a painstaking process because there's, I believe, 14 or 15 nets open at a time. So for 2 people, it does take a while, but there's other folks helping us close them. And there we see Kat tucking in a piece of the net just to make sure it's wound as tightly as possible.
So one thing that we learned is when you're furling the nets, we're always furling towards the gate. And in this way, everything's done in a uniform fashion.
And then from talking with everybody at the station, specifically Katie, we found out more about the birds and their habits, giving us a better idea of what species we should start to focus on for our project. We selected our species based on several factors, including how frequently the species are caught and their behavior and migration habits. With our data, we we're able to confirm the abundance of each individual species during specific times of the year, and then graph their annual visitation patterns. Here we have what's called a GG plot that's produced by R of a Buick's wren's annual abundance patterns.
The precipitation data also came as a file for the years 1998 through 2019, and those were supplied by the National Oceanic and Atmospheric Administration, or NOAA. For the most recent data, we had to go to another website called the Underground and stitch the files together. With that information, we are able to get monthly totals of precipitation to compare. We had the idea that precipitation might have a delayed effect. So with that in mind, we looked into lag periods for our data. Lag is how we show on a graph the delayed effect of precipitation. So after looking at several different resources, we settled on looking at both 1 month lag and 12-month lag.
And Katie provided us the field station data for the birds that we were interested in. And for the last half of 2020 and up until June 2021, we had to hand enter the data into an Excel so we can have R read it as a file. We also analyzed individual birds for p-values by inputting the data into R. And we'll go over p-values more in a later slide.
So these two graphs are showing the abundance of the California towhee over a monthly period from 1998 to 2021. And then here on this graph is a yearly period. So the dots represent the total captures basically shown for the month and the year. With R, as you can kind of tell, the graphs aren't the prettiest. So what I ended up doing was taking the information that R provided us and inputting it into Excel to get a little bit cleaner of a graph that showed the annual abundance patterns, or basically their migration habits. So this is a quick overview of the process. Here we have the R graph, and what these dots are actually providing are a summarized number here. This is an average of all of those dots essentially. And I input the average into an Excel graph, and what we get is this one right here. These particular numbers are all for Buick's wren.
These are the graphs that were created through Excel using the average captures per month for the thrush family. On the y-axis, we have the average number of birds caught, and the x-axis are the months showing 1 year in total. The American robin on the top left is our resident species. And because we have such a limited sample size of the robins, there's no obvious breeding season. Their actual breeding season extends from April to July. If you look at the numbers on the left and then compare them to the other graphs, you can also see that the scale is a lot smaller.
For the Swainson's thrush here in the upper right, we confirm with our graphs that this is indeed a migratory bird. They begin showing up starting April and are essentially gone by June. We really enjoyed these graphs because they visually confirmed the different patterns that each bird has. The last graph here on the bottom is our hermit thrush. It's the wintering species. As would be expected, it winters at the station, and we can see its major population spike when most of them arrive in October.
Migration habits are common among birds, and the specific reasons vary for each species. But in general, birds migrate with seasons from area of low resources to areas with high resources. The two main resources that birds look for are food and nesting locations. As winter approaches, the availability of insects and other food decreases, so birds move south. At Coyote Creek especially, there is year-round riparian area, which provides a relatively stable climate and access to water year-round. There are more available resources around the field station as opposed to other areas, essentially meaning that the birds will go where it's easiest to survive.
This group on this slide is our warblers. The common yellowthroat on the top left is our resident. The yellow warbler on top right is our migrating species. And on the bottom, we have our wintering species once again. It's called the myrtle warbler. There's actually a little typo in there. It's just supposed to be myrtle, not myrtles. We have a greater amount of species of the common yellow throat, meaning you can actually see an obvious season, a breeding season for them, unlike the American robin. Also in July, if you see that abnormal dip there, that's totally normal. It's not them going away. It's just really slow in July, which was actually one of the months that we were there. So we can personally attest to that.
And as you can see from our graph, the yellow warbler seems to show up at this station in large numbers, starting in late summer and early fall, and they depart before winter hits. And the wintering species, Myrtle's warbler, is here starting around fall, and its peak numbers occur in March.
So this is the heart of all of our research that right here. On the left, we have a table showing the p-values for all 18 of our species for both 1 month and 12-month lag. The different colors represent our different species groups: goldfinches, sparrows, thrushes, and warblers. The ones with no color at the bottom are the resident species with no comparison species. In the end, we decided not to use the finches as a comparison group due to the fact that the American goldfinch hasn't really been seen at the station at all over the past few years.
P-values, or probability values, help us see whether or not there is a correlation between two sets of data. It's a value between 0 and 1, where smaller values have a greater significance, which R gives 3 stars, and we outlined in red so it's easier to see which are the most affected. We only used values with greatest significance, so anything deemed insignificant with less than 3 stars would not pertain to our findings. When we input our total monthly bird capture data with total monthly precipitation data, we asked R to make a summary of its analysis, and it will provide us with either a positive or negative number. In our case, a negative means the effects would, or negative effects would mean that months with less precipitation mean more birds were caught, and positive means less precipitation with less birds being caught. The positive and negative effects are not determined by the p-values.
On the left, we have a table showing the significant ratios of our resident, wintering, and migratory species. These ratios are the numbers of birds that showed a significant effect out of the total number of birds in that category. So for example, we see in the 1 month lag section right here that our resident species, only 1 out of the 10 total resident species showed a significant effect, while 4 out of our 5 showed a significant effect in our wintering category. Once again, I just want to mention that lag was a way that we were looking at the delayed effect of precipitation.
And I forgot to mention NS on our graph just means not significant. So precipitation does seem to be related to the number of captures, but we cannot definitively say that precipitation is the sole cause for a change in population. This is a case of correlation versus causation, meaning that a correlation between two variables does not automatically mean that the change in one variable is the only cause of the change in values of the other variable. Causation indicates that one event is the direct result of another, and we can't say for sure that precipitation is a cause of any population trends. It could be one of many causes, or maybe it has nothing to do with it. What we can say is the numbers are highly significant or insignificant with a positive or negative correlation when analyzing the data.
Here we have our GAM model graphs showing 1 month lag for both the American robin, a resident species, and the Wilson's warbler, a migratory species. The y-axis here is a measure of the standardized capture rate, which represents abundance, while the x-axis is the years. The p-values from the previous slide tell us for 1 month lag that yes, the American robin is actually negatively affected by lack of precipitation. This went against our hypothesis of resident species being less affected than migratory species, unfortunately. Upon further research, we found a reason for this, but we'll go over that more in our discussion section later. One thing to note also, there's been virtually no American robins caught at the station within the last couple of years. The p-values over here on the right tell us that also that the Wilson's warbler is being negatively affected by lack of precipitation, same as the American robin. And since the Wilson's warbler is a migratory species, that follows our hypothesis.
And these graphs show two of our species that are not in decline, and they are both residents, the Buick's wren and the California towhee. As with the previous graphs, the p-values show no significance, meaning that they are not affected by the lack or presence of precipitation according to our results. These species are not impacted by drought and are doing well. We think because they are residents, so they are more tolerant to drought, and they also live near water year-round at the station.
So ultimately, bird populations have been declining in general. We cannot say with certainty that populations in our area are decreasing due to lack of precipitation. We were, however, able to find very clear population patterns for a resident, migrating, and wintering species. It wasn't anything groundbreaking, but it was amazing that our data so strongly confirmed that these patterns that other scientists and bird watchers have observed. And thanks to our cutoff for an extremely high significance, we can say that yes, the amount of rainfall did have an effect on some species.
There are some birds that did not entirely fall into our predicted patterns of wintering and migratory birds being more affected than resident birds. For instance, the resident American goldfinch shows a very strong positive effect with 12 months lag, meaning there was less precipitation and less birds caught, while most other residents do not have that pattern. But in general, there was a pattern of resident species populations being less affected than the migrating and wintering ones. Another resident that didn't fit into our projected pattern was the American robin, and we may have found that answer for why they're not affected, and we'll go over that shortly.
Our final conclusion is somewhat general, but it's what the numbers are telling us. Of the species that were affected by rainfall, some of those species are decreasing in numbers. And of the species not affected, some of those species are increasing in numbers. As we mentioned, one of our conclusions was that lack of precipitation seems to affect migratory birds more than resident birds. According to BirdLife International, the State of the World's Bird Report, at least 40% of the world's bird population is in decline. The alarming drop can be linked to industrialization, commercial agriculture, air pollution, and other detrimental factors. Migrating birds would have more opportunities to be affected because they visit multiple areas, and they also have to have the struggle of finding breeding habitat that can potentially be less than ideal. As they migrate, stops along the way may not have enough resources for them. So in the end, they may not even make it to Coyote Creek.
Another reason that our resident birds might not show as much effect as the migrating and wintering ones is again due to the fact that Coyote Creek has year-round water supply and a riparian environment, which offers another level of stability. No matter how little precipitation occurred in any month or year, the creek has never dried up.
So finally, we come to the fact about the robins. One really interesting thing we found while doing our research is that robins, despite being one of our resident species, showed a drastically negative effect to lack of precipitation. What we discovered is that the robin's diet consists largely of worms. And when it's dry, the worms tend to go deeper or entirely die out. And this makes them far more susceptible to drought than seed and other insect eaters. Worms need a moist ground, so the robin's primary food source would be scarce in a drought season.
And we still don't have an exact answer because attributing rainfall as the only reason to population changes would be neglecting countless other factors like habitat destruction, pollution, human interference, and competition from invasive species. More research would need to be done on other potential factors before we can draw conclusions on our project.
So during our time at Coyote Creek, we used a couple different apps, and these are a list of them here.
iNaturalist is a citizen scientist app where people can upload photos of plants, animals, and insects that they see in their day-to-day life. So by the end of our project, we had a total of 83 different observations and 62 species identified, with many of those being considered research grade, meaning they can be used by other scientists for their research. BirdNet is another app we used frequently to identify a bird call when we weren't able to see the bird itself. This app is also interesting because it gives a certainty rating, so we can use our own judgment when an ID is offered to determine if it's accurate or not.
The last app we used frequently, I used a lot before coming to the banding station. It's another identification app, but it's aimed more towards plants. One of my favorite things with this AMP is identifying edible plants and sometimes sampling them. Coyote Creek had quite a few, including some wild radish and elderberries, which are only okay after you cook them. The coolest thing of all the apps we mentioned is that all of these connect scientists with regular day people who just love nature. And this opens up the world of research for both groups. So we're both really going to miss the station. We're both also interested in continuing, hopefully in between our schooling though. And if anybody has any questions, it's hopefully we're not running out of time.
Awesome. Thank you both so much. It was really wonderful to hear about your experience, not just, in the field with the birds, but also thinking about scientific questions and getting to analyze data. That's amazing experience that you've gotten here. So thank you both so much. So as a reminder to everybody in the audience, if you have any questions or even any comments for Kat and Erin, please feel free to enter those in the chats and I'll be reading those out. And I guess first I wanted to start off with a question. So Kat, you mentioned that one of your favorite birds that you caught was the bush tit. Erin, how about you? What were some of your favorite birds?
My favorite bird is the California towhee, just because they kind of remind me of the chickens that I used to have at home. They're mostly ground birds and they kick around in the leaves.
Nice. And have you, I feel like I have heard before that we typically don't get a ton of towhees at the banding station. Were you able to get a few there?
I think in our time we maybe got 2 or 3. I did get to release one, which was the highlight of my time there.
Nice. And then we have comments from Jean, who said, she's really proud. You have accomplished a lot in these 8 weeks, which yes, absolutely you did. So congratulations on that. And then we do have a question, I think, that just came in. Let me see. Oh, just a lot of, great job. Kudos to both of you.
Thank you, everybody.
Yeah, thank you all for being here and spending the afternoon with us.
Nice, yeah. And then also I do want to invite Katie and Jean to also say a few words if you want to. You should be able to unmute yourselves if you'd like.
Yeah, I really like the fact that you did include our first project idea because we were so excited about that. You have these visions and then reality hits. But yeah, kudos to not letting that get you down and also for plunging into some heavy duty statistics.
Yeah, the Rundo was definitely a disappointment. Erin, every time we went back, she tried to take out a chunk of the root and it just kept coming back.
But I don't want Kat and Erin to convince you that they didn't make progress with that, because even though it didn't turn out to be their main project, they went into that with so much energy and took out so much of that giant horrible cane. We have this massive pile of the cane they took out, and it is growing back, but there's also less of it than there has been in 20 years. So they've given us a hope at least keeping it at bay, which is actually a major accomplishment.
Yeah, I want to come back. I want to take it all out. Like, I hate that plant.
Yeah, I'm the same way. One day it'll be gone. One day.
I can respond to a question from Jennifer asking about if the research will be available for scientists. The students have the hope to submit their project as a poster to the Ecological Society of America. We don't have a plan yet on how that would happen funding wise, but it's taking place in Montreal.
That's awesome. That's really exciting. You'll be able to share it with an even broader scientific community. That's fantastic. And then let's see, Gina asked, how does the draining of the Anderson Reservoir affect the bird populations and patterns that you observed? Do you happen to know anything about that?
I can definitely comment that it took out some of their nets. I would say, I don't know how it affects it, but the draining, as I was mentioning when I was talking about the overflow, the way it's kind of situated is almost like a basin. So when it gets drained, it tends to drag everything from up creek, down creek, which just spreads invasive plants, mainly a lot of thistles, which the bees liked, so I'm like, I don't totally hate them, but unfortunately it's not native California plants. So that's the main issue. I don't know totally how it affects the birds. I don't know how long the invasive plants have been around, but that's actually a really good question.
And also thinking about how you showed that the resident species seem to be buffered from these drought effects by the fact that they have some local water. Any kind of removal of a source of water like a reservoir is potentially removing some of that buffer. So that's always something to think about.
Awesome. Thank you all for those great answers. And I guess, let's see. I know that you both mentioned that you have an interest in volunteering potentially, between the school and things like that. Erin, you said you were interested in potentially doing more work at the banding station, right? And then Kat, you said you were interested in doing more habitat restoration. And then with those, would you be trying to, continue working at the banding station specifically, or do you think you'll also be looking for other opportunities outside of the banding station?
I think I would like to continue working with the banding station, but also look for opportunities to work with raptors, because on our last day, I think it was a red shoulder hawk that we caught. And it was really fascinating to see how different raptors are compared to little sparrows and things. So I would like to explore more in the world of raptors and their conservation.
And yeah, I definitely want to continue there just because it's in my backyard and I really believe in what they're doing. And I really, really like the people there. I want to support them any way I can. So we kind of got like I've adopted you now almost. But I want to explore more internships and different types of internships. One of the women there, Jane, she actually introduced me. She did an internship in Antarctic at in her 60s. So I was really impressed by that. And I'm looking into just trying to do as many internships as possible, basically.
Wow, that would be amazing. And then Jennifer also said to make sure to pay attention to the Golden Gate Raptor Observatory site just over the Golden Gate Bridge this fall. They do a lot of raptor work, so that could be a great resource too.
Thank you, Jennifer. I'll definitely look into that after this meeting.
Nice. Well, I think that is all the questions that are coming in right now. So first of all, I do want to thank you both for all this great work that you did and sharing your experience. I think it's really inspiring even for people who have been doing science for years to get re-energized by your enthusiasm. I thought it was just fantastic. You both did a great job. So I guess with that, I want to ask you both, is there anything else that you want to share with everybody before we sign off here?
I just wanted to give a personal thank you to Jean and Raida for setting this up, and then definitely Katie. I know me and Erin both, we really appreciated this opportunity.
Yeah, Kat took the words right out of my mouth. Thank you for everyone who's made this internship possible. Yeah, I'm just really appreciative of this opportunity.
And thank you to SFBBO, because it would have been such a different experience, because collecting 8 weeks worth of data and looking at it versus having decades and decades of data that has been so consistently and carefully taken by volunteers and spent so many hours. That's what made their work possible.
We collected 4 folks to find out things like you guys did. So I mean, appreciate the thanks, but also not necessary, because you're doing exactly what we want. And as Serena said, just making us happy with your enthusiasm.
All right, thank you so much, everybody. Thank you again, Erin and Kat, for your presentation. Thank you, Katie and Jean, for being here and for being fantastic mentors. And thank you, everybody, for joining us. We hope you have a great rest of your evening, and we hope to see you again for a future event soon. Have a good night.
Thank you.
Bye.